Okay, we will remove it. Uh, uh, Good morning, one and all present over here. I can see a lot of familiar faces joining us in person and both online. Uh, I welcome you all for today's Entrepreneurial Talk series. So, for those who have joined us for the first time, let me give a brief about what CDFD is all about. So, BRIC Center for DNA Fingerprinting and Diagnostics is a life science research institute under Department of Biotechnology, Government of India. It is equipped with state-of-the-art infrastructure to enable cutting-edge research in frontier areas of modern biology, including microbial pathogenesis, human genomics, bioinformatics, and others. And CDFD Technology Incubator is a life science focused business incubator hosted at CDFD, where we support biotech innovations and startups throughout their entrepreneurial journey by providing them access to infrastructure, mentoring, fundraising, IP, and technology transfer support. Uh, for those visiting us for the first time, please drop by at our incubator, which is on the sixth floor. After the talk, we would be more than happy to show you around. And since BIRAC's Biotechnology Ignition Grant is currently live, I'm sure a lot of participants who have joined online or in person must be busy with writing or refining their proposals. Remember that having a great idea and a good proposal is just the beginning, but choosing the right incubator makes all the difference. So if you are looking for an incubator for letter of intent, please write to us. We would be more than happy to help you in your big application. Now, coming to today's event, after the success of the first four editions of the Entrepreneurial Talk series, we are excited to bring you with its latest edition. Today, we have the privilege of having with us Dr. Amresh Panda, who will share his journey and insights on how science, innovation, and entrepreneurship can come together to drive meaningful progress. It is my pleasure to introduce him to you. Dr. Amresh Chandra Panda is a leading RNA biologist who earned his PhD degree in biotechnology from BRIC NCCS Pune, followed by advanced research roles at premier US institutions including NIH, University of Miami, and others. With 60 plus scientific publications, 6,700 plus citations, and an H index of 33, Dr. Panda ranks among the top 2% scientists worldwide for 5 consecutive years by the Stanford University. His impactful discoveries on circRNAs, muscle regeneration, and insulin biosynthesis have appeared in top tier journals such as Nucleic Acids Research, Genes and Development, and RNA Biology. He also contributes actively to the scientific community as a reviewer and editor for reputed journals. Driven by a mission to make India self reliant in bioscience technologies, Dr. Panda and his team at BRIC ILS, Bhubaneswar, developed a low cost, high performance RNA isolation kit tailored for Indian researchers. To further accelerate translation of innovations from lab to market, he also founded a company called RNA Biotech Private Limited, a startup which is currently incubated at DBT ILS Bioincubator. He also serves as a non executive director for the same. The startup has already 6 products in the market with a strong IP portfolio of 2 design patents, 2 trademarks, and 2 patents filed. The startup has also secured funding from Startup Odisha and Startup India through Rich Hyderabad to further scale the innovation. And with that, without further ado, let's welcome Dr. Amresh Panda as he takes the stage.
Thank you, Divya, for this nice introduction, and thank you, CDFD, for having me here. So, I'm from ILS. I think he already said a lot of things about me. I had some slides. I thought, like, I will introduce myself with the pictures and numbers, but he did it. So, I'm from ILS Bhubaneswar. So, it's a sister institute of CDFD. So, we are all under BRIC now. So, I think many of you have not seen ILS. So, this is how ILS looks. This is the main campus. The middle building is the research building. Yes, Bhubaneswar. Yeah. So it started in 1989, and now we are here at 2025. It's a small campus. Like yours, we have multiple campuses, actually, three campuses. Says one mostly for research, one for the residential campus, and the bio incubator for having the startups. And there is one also a small satellite campus, or like 2 hours away. So similar to CDFD, we have similar number of faculties, students. I think it is more or less similar to CDFD with more than uh, 800 publications, 36 patents, and uh, products. There are few products which went for the commercialization. Okay. Few products went for commercialization, and few technologies went for startups, and one of them is mine. So before I go into the startup thing, I will. This is not moving. Okay. Yeah, it's working. So similar to CDFD, we have virtual branches which works on different fields like cancer biology, infectious disease, plant biotechnology, and some people like me who are mostly on the technology side, they are m- coming under the interdisciplinary biology because they do not work on a specific disease like infectious disease or cancer biology. So, and the most of the research is done by the PhD students, and we always encourage the JRF from ICMR, DBT, CSIR to apply for the PhD program and then join as the PhD students. Apart from that, several projects also host the project JRFs, but those are very rarely converted into PhD, and that happens only if they have some kind of JRF. So so all the research happening at ILS is mostly through PhD students and few postdocs. And Bhubaneswar, as you might know, it's a planned city. It's a very beautiful city. It's small but very vibrant, and it is also called as the temple city, having close to 1,000 temples in a small city. And I think Dibya already said this. So I joined ILS in 2017, towards the end of 2017, October, and started RNA Biotech in 2021. So it's like 4 years through the startup journey. And he has already said all this. I will not take this thing. And this, my research group has produced 4 PhDs and supported by intramural funds from ILS, Welcome DBT, ANRF, and DBT, extramural support. I will not talk to you about the science we do today, but I will talk to you what general research we do. And this is the group scenario. Currently, we have 6 PhDs, one running extramural grant, one project fellow, and one lab tech. So my lab works on one of the non-coding RNA that is called circular RNAs. So these are the RNA molecules produced from mRNA which do not have any 5' or 3' end. So they are basically single stranded covalently closed RNA molecules without any 5' or 3' end. Because they do not have the ends, they are very stable. And these RNAs are produced from mostly from the exons, and some of them they are generated from the introns or intron lariates. And the lariates, if they are resistant to digestion of the debranching enzyme, they remain as stable lariates and called as intronic circular RNAs or CIRNAs. And these RNAs are mostly detected by two methods. These RNAs you can detect by PCR or through RNA sequencing. So in regular PCR, we put primers facing towards each other. Those are called convergent primers. But when we want to amplify RNA circles, you have to put primers facing outwards. So those are called divergent primers. So in case of linear RNA, the divergent primers will never meet each other. They will run off and will never generate a PCR product. But when they are on the circular RNA, you might get a product, and that can be sequenced to verify you are actually amplifying a RNA circle. And the other method to identify them is the RNA sequencing. That's the first method how people identified circular RNAs. In case of linear RNAs, you all might be knowing we take the RNA, fragment them, sequence, and align with the genome or the transcriptome to get the gene of interest, whether they are upregulated or downregulated through differential expression analysis. But what happens in case of circular RNA is that is different. The reads which align with the genome or the transcriptome directly are of not much use for circular RNA identification. So the sequence which is important for RNA circle identification is the junction sequence reads. And those junction sequence reads come from the fusion reads, which are usually 5 to 10% of the total reads we have in the RNA sequencing. They are aligned in a different manner. As you can see here, the reads are split into halves, and then the first half of the read aligns to the downstream sequence, and the second half of the read aligns to the upstream sequence, which is unusual for the linear RNA reads. And based on this split read alignment, the program gives you a tentative sequence which could be coming as a circle by joining the end of this exon with the end of the upstream exon, start of the upstream exon, and that makes a circle. Correct. Right. Right. How big? Okay, so in majority of the cases, 90% of the circular RNA in any of the cell, they are below 1000, or maybe 80% of them are below 1000 nucleotide. But there are some which can be like 20 kb, 50 kb long. No, because RT-PCR only detects your backsplice junction, not the full length sequence. The full length sequence, you have to do a rolling circle amplification method. We have published a methods paper on that, how to make identify the full length sequence, because sequencing will tell you, okay, this circular RNA have 3 exons. but that may or may not be correct, because sometimes this exon might be spliced out and you will be having only two exons. So that can be uh, identified by rolling circle amplification using RNase H minus RT, so that you get longer cDNA and in a rolling circle manner on the circ RNA, so that you have repetitive full length sequence, and that can be sequenced to get the actual sequence instead of relying on RNA-seq. Right. Yeah, yeah. So that cartoon is showing how the PCR works. So the divergent primers we put, that is in respect to the linear RNA, because it is coming from the parent RNA. So the primers are divergent in terms of the parental gene, but when it comes as a circle, because it joins together, so it will be convergent for the circular RNA. Yeah, going this way. So I think this is clear or confusing? Confusing, right? Okay, so let's see if we can do sequencing of CDFD Hyderabad. Okay, so I imagine this is your RNA or your transcript of interest, and you did fragmentation and then went for NGS sequencing, then alignment. So all the fragments you see, they have aligned nicely. This is how the sequencing works, actually, the analysis. But there are fragments which do not align with the linear sequence, like CDFD center, because this is not there in the linear transcriptome. Okay, so this can only happen if CDFD joins with the center. Okay, and it should look like this. So now, instead of reading Center for DNA Fingerprinting and Diagnostics, I can read from Diagnostics CDFD Center for DNA Fingerprinting. And so the meaning is different. So the same way, when a linear RNA makes a circle, although the sequence is the same, there is no start end, the meaning will be different. So it will not code for protein, but as it is stable, it has other functions, interact with other biomolecules to regulate gene expression. And no, sorry. So the first thing is sequencing. From the sequencing, we can identify circular RNAs, and from the sequencing, we can predict the sequence of the circular RNA, like the start end. And then that start end and the whole sequence the exons in between that can be taken as the mature sequence of circular RNA, NGS sequencing. Okay, for any gene, if you want to test that, you can put divergent primers at different place. No, no, converted to DNA. This is regular sequencing. Adapter based, of course, NGS. The same way we do sequencing for mRNAs, but only difference is the analysis is different. In linear RNA, you take the reads which are aligning with linear transcriptome. Here you take fusion reads which do not align with linear, but they are aligning. Exactly. Like gene fusion, this is done, but from the unmatched, partially matched read. Right. From partially matched, you have to see whether the ends are coming back. Yeah, so there are like 10, 15 programs now. So they can say, okay, first half is matching to the downstream sequence, second half is on the upstream on the same gene. That is the difference. So you did based on that, you find the sequence. And from the sequence, you can take the first half, like 100 base from the last, then join to the first 100 of the start sequence and make the PCR template and design the primer.
And now people have found in probably Homo sapiens all potential
Circular.
circularization, because obviously it is coming from a linear gene.
Linear gene.
So
Yeah.
it is some way there is an enzymatic process that it gets
Splicing.
So that is what the things are there. So in that, how many of such instances have been
That
reported?
more than a million circular RNA have been identified in humans, and in it is like ubiquitously expressed. All eukaryotes have this. So not all genes produce circular RNAs. Maybe 25 to 30% of the genes produce circular RNA, but each gene can produce multiple circular RNA. That is why the number is so big. But the question is how many of them are functional. Many of them are very low abundant. However, there are lot of circular RNA which are as abundant as mRNAs. So we study in the lab like circular RNA which are mostly abundant. And two systems my lab look at, one is the muscle regeneration and the other one is looking at pancreatic beta cell insulin production and how these RNA circles regulate these two things. That is what my lab does. So apart from the basic research, I will not go into the research part. Okay. So apart from the basic research, ILS have a bioincubator. So like yours, the same incubator, and Divya has been a part of that. And Divya was one of the founding members of my startup. And wherever the startup is, the most of the credit goes to him because he was the first person to take it forward and start uh, doing things and getting the product going to market. Yeah. Thank you, Divya. And ILS have yeah,
The largest honest,
one is 100. One startup got 100
that's all.
and others have small amounts. There is one another one he got 3.5 share recently. So I think that is not included here. This was a old slide. Minimum can be 5 lakhs also. Not investment is not 5 lakh like the grant. Okay. Okay.
So minimum,
Maximum was 100. Minimum, I don't know exactly. But I know another person getting 3.5. But the minimum, I don't know. My startup got 30 lakhs as a debenture, not. Uh, yeah. uh, uh, from Startup India seed funds. So that was not angel investment, but that was investment through as a debenture. So ILS has three startups. The first one was RNA Biotech. We started in 2021. Then Polyorbit, another startup, and Bionox is the startup from Dr. Ravindran, who was former director of ILS. He started last year. So there are three companies coming out of ILS. And this one is my startup started in 2021 with a simple problem. Like when I came back from US to India, we saw we are getting all the RNA kits from Thermo, Kaizen, Sigma, and this thing. So, but there are issues with RNA isolation because they all depend on the column-based kits, column-based RNA isolation. So if they give you 50 reactions, you are stick to 50 reactions only. And the column-based things do not isolate the small RNA because when you spin down, anything less than 200 bases will go in the flow through. And also the genomic DNA contamination, because genomic DNA, most of them, they recommend, okay, go for DNA digestion. Okay. So looking at this, I thought like, why not we create a RNA isolation kit that was easier to operate and scalable. And what we did, we started working on the magnetic bead based RNA isolation and developed a kit where you can isolate total RNA, including the microRNAs. And it has the option, if you only need the long RNA, you can still do that. And if you need only microRNA, the same kit can give you the microRNA part as well. So with this technology, we check that with other uh, um, players in the market and compared how our RNA coming from the in-house kit is compared with the other kits. And what we found is it is similar to most of the kits. And when it comes to the small RNA fraction, the magnetic beads are definitely better. So with that, I had two options, either patent it and sell the technology or make a startup. So with at that point, then I thought like, okay, let me publish uh, most of the data, but not all the data, which can be like potentially going for the product development. And it was a MOU signed, and ILS had, okay, I can say what percentage, right? Okay. So ILS gave the technology with an agreement saying getting 10% profit if the company makes profit from this kit. So 10% will go to ILS. With that MOU, I got the technology transferred.
But what is the magnetic that's capable of binding only RNA and
Yeah, so
it has to also bind all kinds of RNAs.
no, see, the magnetic beads are silica based. So the columns also, they are silica coated. Silica layers are there, which is binding with pores for the flow through to go out.
Okay.
In case of magnetic beads, you have beads coated with silica. So it doesn't differentiate between a long RNA and short RNA. So you can bind to all RNAs.
So all nucleic acids, only that you get rid of the DNA first.
Yeah,
then
DNA you get rid of first so that it is not there. So whatever is remaining, you can proceed. Yeah, procedure is I will come to that. Okay. So with that, I thought of starting the company as a startup. Then the question comes with the policies. As a faculty in a government institute, how do I start a company? So then I went to our office and then wrote an application, and then we come through this National Innovation and Startup Policy. I don't know how many of you have read this. So this has clear guidelines, but when I started, DBT had a standing guideline to allow faculties to do it, but the details, how you allow someone to have a job and do a startup, that was not very clear. It was up to the institute to decide, and it took some time, but office finally decided to allow me with certain terms and conditions. Okay, and according to this startup innovation policy, the major guideline says faculties can take 20% of their time for making a startup, or like I come here under that 20% time. Yeah, I think this is the same. Yeah, this was not
The
the
earlier one which you showed was 2018.
19. Yeah,
19.
that was not DBT guideline.
Yeah, it says DBT. It says DBT.
Huh. Yeah, so now it is updated.
Yeah, now it is updated by
25
break.
guideline, it is updated. And the institute can negotiate to have how much share they want to have based on technology. For me, ILS never filed a patent.
So
So
this
I
that
only
you're
had
showing,
the technology know-how.
this is 2025.
No, this
This
was
is also
previous one.
2019.
Yeah, this is previous one. I have never filed a patent for the RNA kit. Yeah. So I only had the technology. So which was transferred like a know-how. So I had to give 2% of the equity. But the guideline says it can go to maximum of 9.5% based on if institute has spent 5 lakhs on my patent and all things, then they can always go for a higher stake in the company. So it depends on how much negotiation the faculty is having with the institute and all that. And
You have identified position for DPT ILS? I mean, in keeping in mind that, have you kind of formalized that this is how we will go as a DPT ILS for other people to follow? Right now, you approached it with the previous policy and other things. Now the DPT gave a guidelines, but again, that guidelines will also need to be adopted by the institute in saying that how would they work it out their things. So like right now you have mentioned 9.5%.
no, this was the guideline. And no more
This
than
was mentioned in the 2019 guideline.
yeah.
Yeah,
Mm.
yeah.
But now also nobody can go beyond this 9.5 party to the company. So no institute would like to have more than 10%. Otherwise, like it will be a problem for the institute if anything happens. Okay. So as you know, ILS, India is having lot of startups. This is the old data. Now I think it is more than 1 lakh startups are there. But they all may not be on ground, but on paper there are certainly more than 1 lakh startups. So this is the startup we started in 2021 with a vision to provide affordable molecular biology kits using magnetic bead based system for DNA and RNA isolation. And as I said, the funding is the first challenge we come through when we start a startup. The first thing who puts the money is the founders or the directors, and then your family or friends, and then the government grants. I will come to that. So most of
Much better ecosystem. So in US, why people want to still, it's expensive to do that. Why still they want to do that? Because investors are floating around. They are looking for early that thing, and actually, some of the people who I know, they don't have to put their money from their pocket. Okay, and actually, the idea was accepted by the
investor, and then which is doesn't happen in India. You have to put your money. If you succeed, everyone is happy. If you fail, you fail. Yeah, that's how Indian ecosystem works. But there are grants we from DST, BIRAC, MSME. So you can explore. I don't have to go through the details. And from students to faculties, there are different kind of like this BIRAC Sitare grant. That is for the students. That's not for faculties. And like this Maiti and Nidhi Prayas. So anyone can apply. And BIG, of course, now they have made it like it has to be a company. So as individual, you cannot apply. But there are grants from and Startup India also. They have seed funds to support. I will not go through this. So that RNA isolation kit, which we developed in the lab, I took the technology to the company and we launched that in 2022 with our former director, Dr. Ajay Parida, and then the Minister of Science and Technology, Government of Odisha. We launched that. And what this kit does is it's a bead-based system, DNA-free RNA isolation without any DNA treatment, and it is scalable. So if you have less amount of sample, you can the 50 reaction kit can be used for 100 reactions. If you have lot of sample for RNA isolation, the 50 reaction kit can be scaled down to 10 reaction, and then you have lot of RNA. So it is solely dependent on the user. But for the column based things, you cannot do that because you are limited to the column. here you can play around the amount. Yeah. Right. Possible. No, what I said is if you have like 1 million cell, you do not need to use the amount like for 50 reaction kit, we always recommend 5 million cells. If you have 1 million cell, you don't need to take 20 microliter bead. You can be good at taking 5 microliter bead, and other reagents also you scale down accordingly, so that the concentration remains same for the beads. Initial material also you scale down. Lesser the volume of the reaction. No, I think it is opposite. If the volume is less where the beads are more concentrated, you have better chance of getting the smaller ones. Longer ones will anyway bind.
I think what is the bead amount decreases, the beads tend to purify the, bind the bigger RNA because they are having,
No,
practically the bead is more.
no, the bead amount you are reducing, so the reaction volume also you are reducing. You are scaling down everything. The concentration remains same. Concentration is not changing. And about the cost, if you compare with the other kits, you can see the cost is not the lowest, but not the highest, and it is the quality is at par with the thermo or the Kaizen kit you are paying for. So
So here comes
yeah.
all the uh,
Not yet.
and is there a differential deal which you are supposed to offer to the
No, see, what we do is like we give sample kits. If you want to buy, we can give you in 1+1. We cannot reduce the price because it's an agreement with the distributor.
this thing
Colleagues. Yeah.
company? So the more the sale is
No, no, but
sale coming to a special supply.
no, but anybody wants to buy, they have to come through the distributor, not directly to us. So even if you come directly to us, you will be routed through the distributor. So yeah, cost is lower.
Problem is the amount of RNA is in the cell. So some companies supply something called RNA protect. That's something equal that you can think of because otherwise we will not be able to.
No, ideally speaking, if you lyse the cells very quickly, RNAs will not do anything, and RNAs affects only if you are precipitating them. Because when you precipitate, RNAs will come in the precipitate and degrade. If you are using beads or columns, it should not be a problem. If you are doing everything ideal, it should not be a problem.
The guys who are selling RNA protect are just kidding.
I will not say that online.
For Agros gel, for example,
Which one you are saying?
all these 5 kits, are they bead based, magnetic beads based isolation, or is it?
Okay, no, okay. This is for a different purpose. Okay, so these kits you all buy, right? And these kits cost different amount of money, and mostly we buy column based kits. To replace all these kits, what we thought of is having one kit that will isolate your total RNA, microRNA, genomic DNA, gel DNA, and PCR, and also the protein. So this was the proposal funded by Startup India. As I said, they gave us 30 lakhs for as a debenture. We I will not go through this. So this is the kit we are coming up with. We have a design patent granted here and trademark also registered, and another patent is going to be filed for this. So this will do all that thing. It is all magnetic bead based except the gel purification. That we could not do as expected, like we thought it will work, but magnetic beads never worked for gel purification. But other things, it is working well.
But how much it costs, like for kit?
We have
per-
not started selling this.
what
yet.
expected price?
I think, oh, I deleted that slide. We plan to have it like 30,000 per
Or
kit,
how many reactions? Like
50 reactions.
Okay.
50 reactions, so that you have the option. Like you do not need to buy multiple kits for multiple things. So one kit you use whatever you need in the lab. So that is what we plan to come up with. And as I said, like these ones here, like there are similar things in the market, but the bottom one is our kit, which is providing more facilities and at a 30,000 cost, and mostly targeting the research labs and college or universities. And once you isolate DNA and RNA, the other problem we face is the running the gel and visualize them, right? So we all like boil in the agarose in the microwave, pour that, and then run that on the gel agarose system. We need a power bank. And how much time I have, Divya? Okay, so we have a power bank to supply the power for the gel running, and then we take the gel to a gel dock, which is sometimes in our lab or sometimes in central facility, right? So all students might have faced that. Yes or no? Right? So to overcome that, I thought is like we can have one integrated system to do all that. And this is the system. And where you can boil your agarose, then you pour your gel and cast it, and inside that food you have the gel running apparatus. You put your gel there, run it, and you have a camera for real time visualization of the gel and take your image. But now we have removed the USB because we launched this, and some people suggested, okay, USB may not be a good idea because it might corrupt the system. So now we are removing the USB. It will have only Bluetooth so that you transfer your photo to your phone or system directly from there. So it will be an integrated system. We have not started selling it. So, but if anyone wants to order or CDFD wants to order as introductory offer, we can give you at any price you want. Okay, that's my offer. If incubator wants to buy, I will give it at your price. I will not tell you the price. But once it is on paper and we have agreement with the distributor, I cannot do anything. Then you have to buy on the sell price what is there on the website. We have not put a price till date.
Buffer check bar and all that.
Buffer, yes. Initially, we will provide for few kits. This will be regular buffer, whatever you are using. Yeah, you fill it after use, you discard. The same way you do it in the lab. It is not replacing your buffers or anything. It is only replacing multiple instruments sitting in the lab. It is just one instrument and you are done with your gel electrophoresis and visualize. And these are the kits already Divya mentioned were in the gem also for order. And as sir asked, like I'm replacing other kits. I'm trying to, but it's not easy to replace. And the challenge is the competition you have with the players in the market, and these players are mostly the big players and our uh, acquaintance with the kits we are using for years. So that is the whole issue. Like we are having another RNA isolation kit, so people will say, okay, this is similar to any other kit in the market. And the other challenge with the biotech is it takes time, which nobody understands, not the investor, not the government grants also. Government grants give you grants for 1 year or 18 months and they think it IT. That is why I put it here so that people realize it's not easy for a biotech startup to succeed because many things are uncontrollable in biotech and and mostly it is like takes lot of money to start from idea to the final product and the time it doesn't happen overnight, which happens overnight for the IT people and investors and other people. They think, okay, in biotech also you get a product overnight. That doesn't happen. And most of the time, like 90% of the startups do fail, but rarely like 1%. In 10 years, 90% of the startup vanishes for sure. But there is a hope. India is on the third rank on the unicorns. So hopefully our startup ecosystem gets better. Hyderabad definitely have a better ecosystem compared to Bhubaneswar. And I'm here to learn how this ecosystem works actually. And
But not work with your with Indian startup channel. So it is also communicating your success story to other startup. Second, make your project, the kits available at some kind of arranged discount price as a, like, there was many freezer system, that is the thing. So since these are all stable under the, either the roof, some
4 degree.
4 degree. So most of the things are actually possession and don't expire in a very short time. So maybe, Kavita, just think of a kind of proposal, such kind of proposal, which is also cross promoting the innovation at the same time making it available to the starter.
I just wanted to ask about his distribution channels. So, how many distributors do you have across?
We have one in Hyderabad,
Okay.
one in Bhubaneswar. We have multiple.
Okay.
Okay.
One in Hyderabad. Up north?
No.
No. Okay.
Sorry? No, not all components. There are few components which need to be at 4 degree.
So that was a cross promotional plus at the same time making it, see, the one of the problem is ease of access. So
No, sir, the first problem is not ease of access. If you tell me you need a kit tomorrow morning, I can give it to you. You.
sometimes the student is using a thing he does not want to change.
Change. That is the first problem, sir. I was coming to that. Yeah. Ah.
Can get more.
It is not about cheaper, sir. I will never say it is cheaper. Hmm. With quality, it is same quality you are getting at a value. More isolation with the same amount of money. And in the same time, you are supporting the Indian ecosystem. Okay. Most of the scientists probably they do not believe it just because some startups do not maintain the quality and it happens. Okay. And it also happens with the big companies also. I can tell you, but I will not tell you here. But students or faculties do not blame the company. They think it might be a problem with our protocol, things didn't work. But if it happens with a small company, the blame always go to the small company, the reagents are bad. Even if we have stored it differently, which
That is where branding matters.
branding matters, and that needs money and time. Lot of money.
Like cross promotion at the same time having some sort of arrangement that I am promoting, because all incubators are also supposed to kind of that their products are. So those will be available, and once a research lab wants to make use of that, they can access and they can get it and internally build or whatever the things are there. So that is something which is there. I would
That is why I'm here, sir. Yeah. So
uh, I did not discuss it online, but I just wanted to kind of explain about the distributor margins. Okay, so
we'll discuss, sir.
discuss. I don't think it's a third position. That is 5% or all over.
Which one? All over, all over. That is not biotech. Biotech, I don't know how many unicorns you are asking.
Is India in terms of like biotech sector, how does it fare in biotech sector?
Do not have the statistics, ma'am.
But which country is US is leading there also? US is leading.
In biotech,
Hmm? No, I don't think so.
think,
Don't think so.
yeah, maybe. Hmm.
3,000 star in the US.
Hmm. Yeah, but I don't know right now.
So one of just find with all these kids is there isn't a feedback mechanism that operates, especially deal with big companies. Right? I think if you have a mechanism of getting feedbacks, in a very simple way, that would probably, people will get more faith. You say do this, my kid is going to work better. Maybe you made this mistake, and it improves. I think that's going to be the reason why somebody will prefer a.
Correct, sir. But being a scientist and being in a government system, I cannot tell my colleagues to buy it. I have never done that.
No, no,
Okay.
not you're not saying it. I'm just
No.
telling you that
Unless I say that a salesperson saying to another scientist, they do not take it seriously. And as a scientist, I am not going to tell anyone you buy it. Even in our institute, few labs bought it. Most of them do not still rely on thermo. I have never said anyone ki you buy this.
not
Because
not
that is a
not forcing you to buy.
conflict
If
of
some
interest. I cannot tell anyone you buy it.
No, but if some, what I'm trying to say is one way to encourage customers would be to satisfy the customers, problems they face. I mean, you give a feedback. For example, I know that, someone I know who has done a PhD works for in vitro gen, but the job of the person is to clarify to customers, if they say, they should be able to talk some science, right? I mean, I don't know how your
No,
distributor system works here.
no. If wherever our distributor goes, our person also goes. It is not on the
Our
distributor.
person means you're
Means
an academician.
company person.
Uh,
Company person also goes. It's not relying on distributor only route the financial orders and everything. But the technical part is on us, not on the distributor. They cannot do that. But that happens when we have 100 customers, we get feedback, and
hmm.
we do not have 100 customers yet.
But, uh, but I think it's a circle. They get satisfied, they will tell others, and then that's how the customers will increase. Right? I mean, it's like a vicious circle.
Yeah. Okay. So yeah, so this is the last slide for the students and anybody who wants to start incubators. Like this is my learning what from my experience and from knowing the startup system. Like it should be a breakthrough technology and big share of a small market. It should not be like thinking, okay, one technology will disrupt the market. That's not going to be very successful like ITs. This is biotech. So, and also the right team. And you should have some secret like the patent. Like we have not filed the patent, but I have my own secret. So, so other person cannot read my paper and make the kit. Okay. So, and also social entrepreneurship. like what people believe in, like the solar panel business. You imagine, okay, like now AI. It's a social entrepreneurship. People believe in it, so more people are putting money just because of the buzz. Okay, so that you get more money. And the final thing is like if you fail, do you have another option? Okay, and thank you. There is a scorecard also. I will share with Divya. Maybe he can send. There is a scoring. You can score for yourself and see if you are actually ready for a startup or not. If you are not ready, just because emotionally you are thinking you can start a startup, don't jump into that. That's not a good idea. There are clear guidelines whether you should start yes or no. Okay.
So now the floor
Thank
is
you.
open to questions. If anyone has any question, please. Any online participants, you can unmute yourself or put your questions in the chat box.
I have a person who does that. I do not do that. Yeah. Others. Yeah, that is what I do. Yeah, that is what I meant. Like, I'm not telling my colleagues because I cannot tell them. I can tell you.
So he has like obligations as a scientist.
There is something like internal marketing and external marketing. Internally also we need to marketing. External marketing is anyway marketing.
So why
Conflict of interest, because you are becoming
You
an
are
agent of the company in the where you are employed.
employ and suppose I'm having collaboration with you. We are working on a scientific thing, and I'm saying, okay, you buy my kit. That becomes a problem.
Any more questions?
The actual team is 3. Okay, me and 2 more person. There are more people who are on the background, but I will not say they are actively doing things, but 2 people who actually do the thing.
Who are on the
But
payroll.
yeah, and I only give the idea. So for a big team like Sachet, we need more distribution partners and everything. Everything needs money. As I said, startup is all about money. And I do not have that much money to put in to spread the business.
Could
At
you
this.
tell us a
Yeah.
little bit about the grants, government grants that you got?
Yeah, so as I mentioned, Startup India seed fund, that is one grant we got 30 lakhs as debenture from Research and Innovation Council of Hyderabad here. And then we got 16 lakhs from Startup Odisha. And Startup Odisha also gave a monthly support for 1 year.
These are spread over how many years?
1 year.
1 year. 30
Wow.
lakhs and 16.
1 year and 30 lakhs is for 1.5 years. Yeah. So that is what I was saying. Like these grants doesn't last for long.
Commit that this money is going to be used for ABC
Yes, yeah, of course.
target base.
Yeah, yeah. So and they do not give you the whole money at a time. They evaluate
See for financing also have a term sheet.
term sheet.
Budget ahead.
So
Yes, so yes, same like objective based. Okay, 1, 2 will be done within the 6 months. Then you send the progress report, and then this much money is spent, so you get the next one. The same way. Yes, yeah. But only grant that doesn't allow is the Nidhi Prayas. They give you like up to 10 lakhs for developing a prototype, but they do not provide you money for the manpower, salary. But sorry, monthly allowance they have provide 22,000 per month for 1 year. Yeah, employing a person. Yeah, from Odisha government. Yeah, startup Odisha. Yeah.
So right now you are not under labor laws and other, because there are only 2 people.
Yeah, yeah, yeah.
Space technology is completely novel, is it, or there?
No, no, there are tons of during COVID, sir. Actually, the company got late in launching this because of all this regulatory and this thing. No, no. See, this technology is not very new. Okay. Yeah. So, and this making the beads and only the buffer and those systems are very old. Only thing you need to know how to make it work. Okay. But
Approvals you got, regulatory approval.
regulatory approvals from ILS, as I said, like how much time you can give, and in the undertaking there are certain clause like I said, I came here under that 20% time frame. I took a leave from the institute. Okay, I'm going to promote my company. Give me a leave for that. So ILS has that provision for 20% uh, allotment of your time for the company. Product for the research, there is no technical regulation, but if it has to go for diagnostics, it has to be done. We have not done that. Like initially we thought like during COVID, we wanted to have this magnetic bead. Like there were tons of companies, they come up with magnetic beads for, but that regulatory thing needs a lot of money. So we said we are not going there. Already there are 50 companies who are making magnetic bead based RNA kits, but those kits do not work for cells. What we made is an universal kit that works for plant, blood sample, tissue, animal cells, anything you put in, it will work. For any company you go, you have a different RNA isolation kit for plant, different RNA isolation kit for blood, and different RNA isolation kit for microRNA. So we removed all that, having just one kit for everything. End of 2022. RNA kit. Yeah, 2022 actually. But
So how did you manage the directorship registration of the company? Right? So you are not there on the director, right?
I am on the director.
No,
Non-executive.
executive or non-executive?
Non-executive.
Non-executive.
So in it has to be.
In your registration, you have to provide two
Directors,
right?
yeah.
So one is you,
There is another person.
there's another person,
Ah.
right? So directorship is that you have to take do you have to take permission to get it PIN number to your office? No.
No, no, that is a blanket permission for the directorship. Okay, otherwise, why would I need to inform my I office if I am not the director? If I am a shareholder, I don't need to inform my office. If I
No,
put
now the new guidelines envisage that also, if you are taking.
shares also,
Yes.
shares on a company which have intellectual input from you.
Yeah.
Yeah, that is what I mean. Like, and for the intellectual input, we have another agreement. And for this time commitment, I have another terms and condition like 2% of the equity from the company is given to ILS. So these are two different things. For directorship is one terms and condition, and for the technology it is a different terms and condition.
Maybe we can take more questions offline. In the interest of time, we will conclude the session here. Thank you, sir, for the
Thank you.
insightful presentation. Thank you for sharing your thought-provoking ideas about how science and entrepreneurship can go hand in hand to shape the future. Now I request M. Kavita Rao, scientist in charge CDFD Technology Incubator, to hand over a small uh, token of appreciation to our guest. With that, we conclude the session. Thank you, everyone, uh, to who joined us either in person or online. Thank you once again.